Thanks so much, Soumya. That was a really kind introduction. And thank you everyone for being here. Good afternoon. I'm excited to share with you some of the work that I've been doing on the topic of integrating large-scale single-cell genomics data in the context of immune-mediated diseases. So let's get started. So normally, our immune system helps protect us from things like infection and cancer, but sometimes an abnormal immune response can lead to immune-mediated diseases. So these are clinically heterogeneous chronic conditions that collectively affect 5 to 7% of the population and include things like rheumatoid arthritis, RA, lupus, type 1 diabetes, IBD, psoriasis, amongst others. And in all cases, there's pathogenic inflammation that causes damage to our body's own healthy tissues. And so these are complex diseases which include both genetic and environmental components. So to understand these diseases, we really have to understand the immune system. The immune system comprises many different cell types, which are produced as part of hematopoiesis, and they include both innate immune cell types like monocytes, which can recognize many diverse pathogens, and as well as adaptive immune cells like T cells and B cells, which can mount a more specific response and have immunological memory. And while all of these cell types have the same underlying genetic code, or DNA, they read the DNA differently and produce different amounts of RNA transcripts that encode proteins that lead to their diverse cellular phenotypes and functions. And so single-cell sequencing has become a very powerful tool to understand the immune system and the cell types that drive diseases. And the most common method for this is single-cell RNA sequencing, which lets us measure the RNA within individual cells. So starting with a sample of interest, say synovial joint tissue from rheumatoid arthritis patients, we can isolate the individual cells into tiny droplets, reverse transcribe the RNA into DNA, which we can then sequence to get a readout for how much of each gene is detected in each individual cell. And this is all very exciting because previous methods like bulk RNA sequencing tend to average all the cells from a given sample together like a smoothie, whereas single-cell RNA sequencing lets you measure actually each individual cell where you can distinguish each type of fruit or cell, like a fruit salad. Okay. And the resulting data usually takes the form of this matrix, the light. Okay, let me turn. So the resulting matrix, that's better, yeah. Usually takes the form of dimensions genes by cells, where each element of the matrix is the number of RNA transcripts detected for that gene in that cell. And so to process this data, we usually apply some sort of dimensionality reduction, most commonly principal components analysis, or PCA, to then reduce the dimensions from, say, 20,000 genes to 20 or so PCs. And then we can integrate the cells to remove batch effects and then cluster the cells within this coordinate space to get a plot that looks like this. So this is a UMAP plot, which is a 2D way to visualize the coordinate space of all the cells, where each dot is one cell. And so in general, the cells that are closer together in the UMAP are more transcriptionally similar. So you can see here that there are many cell types present in synovial tissue, like T cells and stromal cells. And then we can take this one step further and take all the cells of a certain cell type, for example, take all the T cells, and do the whole process again, finding the principal components that best explain the variation amongst T cells. And in this way, we can define finer grained cell states within a cell type. So here we have many different colors for the T cell states. And in this way, single-cell sequencing is giving us really unprecedented insight into the cellular heterogeneity within samples. And I would argue that at this very moment, we are in the single-cell golden age. This plot shows the number of single-cell studies published across time. This is about when I joined the lab, and so you can see around in the past couple of years, there's just been a boom in the number of studies. And then not only are there more studies, each study is now sequencing more and more cells. So the plot on the bottom is showing the number of cells per study with the y-axis on a log scale. So you can see in recent years, studies with hundreds of thousands to even millions of cells are becoming available. And this is very exciting because now we have a lot of data to be able to ask interesting questions. And a very promising strategy is to be able to analyze multiple data sets together. This can help us do things like boost our power, to compare across different contexts. But in order to do that, it's very challenging because of this reason where there's many different sources of variation amongst the cells. So some of these differences are driven by biological effects, like differences in cell types or cell states. These are effects that we're very interested in. There are also differences amongst different tissues or between individuals, capturing things like age or environmental exposures, or even different species. And then there are these differences that are driven by purely technical effects, And these are usually things we always want to remove or somehow correct or account for. And these can include differences in processing batch, like different personnel or reagents. Different samples might have been handled differently. Data could be generated by different groups as part of different studies, or even sequenced using different platforms or technologies. And so here's an example of what a technology effect might look like. So here we have PBMCs, or peripheral blood mononuclear cells, that were sequenced with 3 different sequencing technologies, which are the different colors. So if you run a simple PCA-based pipeline, you can see here that the cells primarily cluster by which technology they came from, making it hard to identify the cell types. Okay, so now that I've discussed immune-mediated diseases, single-cell sequencing, and a taste of why it's challenging, I'll now introduce the main guiding question of my PhD, which was how do we leverage many large-scale single-cell data sets to better understand immune-mediated diseases? And to answer this question, I think it can be broken down to asking good questions and developing good methods, which is the outline of the talk. So in terms of thinking about what to focus on to study, I decided to kind of focus on the most important region of the genome in relation to immune-mediated diseases, which is the HLA, or human leukocyte antigen region. And then to do this, I had to develop 2 methods. The first is a method to accurately measure HLA expression in single cells, called single-cell HLA pairs. And then the second is a method to integrate multiple large-scale data sets together. It's a method called Symphony. The first method is going to really help us correct out biases that exist in standard pipelines, and then the second method will really help boost our power to integrate multiple data sets together. Okay, so I'll first start by motivating why the HLA region is so important to study. So as some background, the HLA genes are these set of genes that are located within the major histocompatibility complex of the genome, also called the MHC, or HLA, and it's on chromosome 6. And it has a very long history of being studied because it's important in many areas of medicine, like cancer, transplantation, infectious diseases, and autoimmunity. And the HLA genes the classical HLA genes are broken down into class 1 genes and class 2 genes, where the class 1 genes are HLA A, B, and C, and the class 2 genes code for the molecules HLA-DR, DQ, and DP. And in terms of their biological function, the HLA molecules are very important for the immune response. Because their main role is to be able to present antigens to T cells. So antigens are these little protein fragments called peptides, which can come from within or outside the cell. And they're presented on the HLA molecules, and they're presented to T cells, which if a T cell can recognize the HLA peptide complex, it can activate the T cell and start an adaptive immune response. And so for HLA class 1 genes, they're thought to be on all nucleated cells and present peptides derived from inside the cell, like cancer or viruses, to CD8 T cells. And then class 2 genes are thought to be present on antigen-presenting cells, and they present exogenously derived peptides, like from phagocytosing a microbe to CD4 T cells. And so in this way, they serve as the bridge between the innate and adaptive immune response and help the body know how to differentiate between self and non-self. And so an interesting thing about these genes is that they're very highly polymorphic across people, which means that there are many different alleles or versions of the gene across the population. And this makes sense because as a species, we've co-evolved with pathogens, and it's advantageous for us to collectively be able to present many different types of peptides to protect us against different possible infections. And because there's so many different alleles or versions, these different alleles we have a very complicated nomenclature system to be able to name the different alleles. So for example, here's an example allele where the first part is which gene the allele is for, like HLA-B, and then the next parts can be is showing at different levels of resolution how we want to define the allele. So for example, 2-field resolution lets you define different variations among the amino acid sequence. And here's an example crystal structure for HLA-DR that's bound to this little antigen in pink. And so across time, we've discovered that HLA has the most associations with diseases genome-wide. So investigators have done these genome-wide association studies, or GWAS, looking for associations between places along the genome to diseases or traits. And so you can see here on the x-axis, there's the different chromosomes, and the y-axis is the number of associations that have been found to different loci in the GWAS catalog. So you can see that there's just a really high number of associations to the HLA region. And in terms of immune-mediated diseases in particular, HLA has the strongest genetic associations. So typically they have effect sizes larger than all the other loci throughout the genome combined for many diseases. And here are some examples for many diseases associations with different HLA alleles. So for example, HLA-B27 highly increases your risk for ankylosing spondylitis. HLA-C0602 increases risk for psoriasis. Different HLA-DRB1 alleles increase risk for MS and RA, and the list goes on. There's many examples. And exactly how these variations lead to disease or are associated with disease is not fully understood, but there are several possibilities. So the first is that there are coding changes in the body of the gene itself that lead to amino acid changes in the HLA molecules. So that changes the protein structure. So you can see here some example variations in HLA-DRB1. Maybe they lead to changes in the DRB1 protein, and that affects the quality of antigen presentation. So maybe certain alleles are better at presenting self-peptides or restricting the TCR repertoire. And there's been a lot of work that has been studied for this. And then one alternative hypothesis that's not mutually exclusive to the first is that there could be regulatory variation playing a role. So here, instead of the variations happening within the gene, they may not necessarily be within the gene leading to coding changes. They could be potentially upstream of the gene in a promoter or maybe an enhancer that could lead to changes in how the gene is transcribed and regulated. So for example, maybe there's this blue SNP here that might affect the binding of a transcription factor. And depending on whether the transcription factor binds well or not, it could affect the quantity of antigen presentation by changing the levels of HLA-DRB1 transcription. And there's been some evidence to show that HLA expression levels are important for disease. So for example, the two studies here, the one on the left was showing that HLA-C expression levels, independent of the coding variants, can affect whether someone controls HIV infection and also increases risk for Crohn's disease. And then the other study tried to fine map disease risk alleles for lupus and argued that they lie within regulatory regions that modulate class 2 expression. So this brings me to the concept of an EQTL, or expression quantitative trait locus, which is another name for these kinds of regulatory effects. So an EQTL is basically a genetic variant, like a SNP, that controls the expression or is associated with the expression of a nearby gene. Here we have a blue EQTL SNP that is associated with the levels of gene G. And so you can have across the population three different possibilities for the SNP. So depending on the chromosomes that you inherit from mom and dad, you can either be AA, AC, or CC for this blue SNP. And then depending on the dosage of the C allele across people, that can be associated with the expression levels for gene G. So we would call this an EQTL. And typically, traditionally, EQTL studies were done using bulk RNA sequencing, where people try to associate the gene expression levels to the genetic variants across samples. Now, with single cell data, we can now better understand at finer resolution how the EQTL is affecting transcription. So here we have single cell data. We can now separate out the cell types and the cell states in the data. So here's an example of an EQTL effect that's very specific to B cells, but it's not present in the other cell types. Single cell data allows us to be able to dissect this. And then furthermore, we can find dynamic EQTLs that change in their strength across cell states within a cell type. So in this example here, we have T cells that are all T cells, but then if you look across the T cell landscape, there may be an effect where, depending on where you are along this continuous landscape of T cells, the EQTL might have a stronger effect, say, in these darker T cells. And so to be able to understand these regulatory effects in the HLA, their potential roles in diseases, we really need a map of the EQTLs for the HLA genes and their expression. So we set out to systematically map HLA expression as well as the regulatory variants across different cellular contexts. And there have been some previous efforts to do this in bulk, and they primarily have used homogeneous cell lines. So we really thought that single cell data could help us here because we can now assay diverse cell states that come from in vivo contexts and map context-dependent EQTLs in multiple tissues and data sets. And so to do this, we found these 4 data sets that have immune cells that were sequenced with single cell RNA sequencing and all have genotype of data available for the same cohorts. So these include 2 tissue cohorts, one from RA patients, their synovial tissue, one from intestinal tissue from IBD patients, and then 2 from peripheral blood, one from blood cultured in vitro with influenza virus, and then 1 from a large population cohort. And so these, in total, the data set includes 1,073 individuals and about 1.36 million cells across the 4 data sets. And so at a high level, again, in our EQTL analysis, what we're trying to do is associate the different SNPs to HLA expression. So I'll start by focusing on the SNPs part, because that's a little bit more straightforward. And so to get a common set of SNPs across these 4 different data sets to be able to test in our analysis, we used a method called HLA imputation, which allows us to impute HLA variants, so SNPs, as well as HLA alleles, using a large reference panel. So applying HLA imputation, we were able to impute a total of 12,000 variants that were common to all the different data sets, including variants across the MHC, as well as their classical HLA alleles for each individual. So now we have the variants. Now to the other side of the equation, we need HLA expression estimates. And turns out this part was actually a lot more challenging for technical reasons, which I'll get into. So this brings me to the first method that we developed called single-cell HLA pairs, which is a way to accurately measure HLA expression in single cells. So first, why is it hard from a technical point of view to quantify HLA expression with standard pipelines? And the reason for this is, if you remember, I mentioned that the HLA genes are very polymorphic across people. That leads it to be so that they're not very well represented by having a single reference genome. So you can see here all the different alleles for HLA-DRB1, all in a multiple sequence alignment. And so you can see that there's a lot of regions of both structural, so large indels and insertions and deletions, as well as single nucleotide polymorphisms across these different alleles. And so that can lead to this issue where if we're trying to estimate someone's HLA expression, what we usually do in a standard pipeline is take their reads and align them to the reference genome. But here we have this issue where, take for example, we have 2 different individuals, one of whom has the orange triangle allele for DRB1, one of whom has the blue square allele, and say for this example that the reference genome has the orange triangle allele. If we try to estimate the expression for the orange individual, what we usually do is we'll take their sequencing reads, align them to the reference genome, and then count how many transcripts are present for that individual, and we'll be able to accurately measure their expression for the orange individual because the reference genome well captures their sequence content. However, for the blue square allele individual, if we align their reads to the reference genome, what can happen is that there will be reads that, due to the sequence polymorphisms, don't align well and fail to map to the reference. And that can lead to unmapped reads, and then eventually we'll underestimate that person's HLA expression. So this is a big problem because that's like the dependent variable in our analysis. And so to overcome this issue, we developed a new pipeline called single-cell HLA pairs, which stands for quantifying single-cell HLA expression with a personalized pipeline. And the intuition for this method is that we, instead of using a single reference genome, use personalized reference genomes, one for each individual. And so the way this works is we have already previously imputed the HLA alleles for each person, as I mentioned, and then we can go to a database of the sequence content for those alleles and add the sequences to the reference genome for each person, so each person gets their own personal reference genome. And then we align the reads for each person to their own reference. And so by doing this, the output of this tool is still a genes by cells matrix, except for the HLA genes, the estimates are more accurate than running a standard pipeline. And so we did several benchmarking analyses to show that personalization really improves the HLA expression estimates. So here in this plot, we're showing for all the classical HLA genes, each dot is for each individual, the percent change in their estimated expression for each gene. And so you can see that for some individuals, compared to a standard pipeline, which has a single reference genome. And so you can see for some individuals, their expression estimates change by like 50% or more. And importantly, it has to do with how different someone's alleles are from the reference genome. So for example, if someone has the same exact alleles as the reference, their expression estimates don't change much when we personalize the reference. But then for individuals who have more different alleles from the reference, they tend to have higher increases in their estimated expression. And this second analysis really helped convince us further that this is the pipeline is working correctly. And in this example, one of our data sets, interestingly, was sequenced the same cells, but using 2 different read lengths. So 84 base pairs and 289 base pairs. And so we reasoned that these different read lengths could be leading to different degrees or kinds of mapping biases based on the read length. And so we reasoned that if we run a standard pipeline, the long and short reads may disagree in terms of what the true estimate for the expression is, but in a personalized pipeline, if it's working correctly, they should agree better. And that's exactly what we saw, where the top row is showing the concordance between long and short reads using a standard pipeline, and then the bottom is showing using our personalized pipeline, and we see for all the genes, there's improvement in the concordance. So for example, HLA-B improves from 0.82 to 0.97. And so now we have this pipeline that we can use to quantify HLA expression across single cell data. And so here we have the new HLA expression estimates with our pipeline for class 1 genes, and then class 2 genes on the bottom across these different major cell type categories across the 4 different data sets. So as we expected, class 1 genes are highly expressed in all cell types, and then class 2 genes show more variable expression across the cell types and data sets. Interestingly, we did see class 2 expression in cell types that were not traditionally thought to be antigen-presenting cells, so for example, T cells and some stromal cells, so that was very interesting. Okay, great. So now that we've talked about the first method, I'll talk now about the second challenge, which is how do we integrate multiple different data sets together to really do a much larger scale analysis. And so for that, we developed this method called Symphony. And so, yeah, combining multiple data sets can help us do things like improve our power by having more individuals and more cells and validate across different contexts. And this might be a slightly longer discussion, so bear with me. Okay, so to go back to this slide, where we have many different potential effects that may be contributing in single cell data, let's start with the example that I mentioned where we have 3 different technologies for the sequencing PBMCs on 3 different technologies. So here's the PCA-based coordinate space if we don't do any integration. And so for this, so what we want to do is to be able to align this correct cell states with each other across the different technologies and remove the effect of technology. And so for this problem, we turned to a strategy called de novo integration. And there are many methods to be able to do this in the field, including one that our group developed called Harmony. And so as a concrete example for what Harmony does, we can apply Harmony to this example here and then integrate the cells so that now the effect of technology is removed and the cells now cluster by cell type. And without getting too much into the details, Harmony basically uses a soft clustering approach to find clusters that are well represented across the different data sets, but also maintaining similar expression profiles. And the user has to specify the batch variable, in this case, technology. And then it uses a linear mixture model to be able to estimate and then remove the batch effects in the embedding or the coordinate space. And so that by the end of this process, the cells are now integrated into this shared harmonized coordinate space, which can then be used for finer grained clustering or cell type annotation. And so as you can see, de novo integration is a really powerful strategy. It helps us remove unwanted variation and align cell states. It can handle complex study designs, and it's been widely used for constructing single cell reference atlases in the field. However, it does have some limitations. And the main one I want to focus on is that it's difficult to query, and that is it's difficult to add new cells to the reference once you've already made it, because it requires you to reintegrate all the existing cells. And that may cause problems like corrupting the original reference coordinates for these cells that were previously there. And it's very inefficient for very large scale data sets where you have to like send large data matrices across different groups potentially, or rerun large processing pipelines on many cells. And so imagine that you're doing a single cell experiment. You put weeks and months into generating this atlas for a tissue of interest, so say PBMCs. And say you've generated this reference atlas using de novo integration. It has 500,000 cells, so you're really happy with it. And then one day your collaborator comes along with a smaller data set, say 50,000 cells, and wants to know what types of cells they have. One option would be to integrate your cells with their cells using de novo integration altogether. But as I said, that would kind of be prohibitively slow for very large data sets. So instead, there should be a way to ideally quickly map the query cells onto the reference atlas, starting from the query's gene expression values. And this is also useful in the case when you have multiple query data sets and you want to somehow define a standard way to map the cells onto a common reference to be able to analyze them. And this, you can imagine, would be very useful for our HLA study when we have multiple data sets. And so we developed a method called Symphony, which is a general purpose framework to be able to do this. So Symphony comprises two algorithms. The first one is called Symphony compression. And that is an algorithm to take in a harmonized reference atlas, and it outputs the minimal reference elements that are needed to perform the mapping of new data sets. And the second algorithm is called Symphony mapping, which takes in a query data set, its genes by cells expression, as well as a compressed reference that you made in the first step, and then maps the query cells into the harmonized reference coordinates. It approximates running de novo integration on all the cells, but it does not require the reintegration of the reference cells, so the reference remains stable during that process. And in contrast to automated cell type classifiers, which are another kind of method which assign the supervised methods to assign discrete labels to cell types, Symphony is able to map cells into a continuous coordinate space rather than assigning rigid discrete cell type labels. And so here's a visual for what's going on. To build a reference, we'll start with many reference data sets. For example, multiple studies, donors, or collection sites. Here, the shapes represent cell types, and the colors are more fine-grained cell states. And so by projecting the cells into a lower dimensional coordinates using PCA and performing de novo integration using Harmony, we can now get the harmonized reference coordinates where the cells group by the cell types with the unwanted technical and other variation removed. And so this is where Symphony comes in. And so Symphony compression takes in this harmonized reference atlas and it computes the minimum information that's needed to map new cells. And as I mentioned, Harmony uses this underlying linear mixture model where each cell is potentially assigned to multiple soft clusters. And each cluster learns its own model for how to remove the unwanted effects. And at a high level, what Symphony is doing is capturing summary statistics on each of these models, so we don't have to save information on all of the cells together. And then the next part of the figure shows how Symphony mapping works. So here, starting with a new query data set, the red cells here, where we don't know their cell types, we can then map the query cells into the reference coordinate space, and then assign them across the soft clusters defined using the reference, and then assign the appropriate mixture model components to each query cell to be able to then remove any batch effects within the query. And then by the end of this process, the query cells are now in the same integrated coordinate embedding space as the reference cells. So here we have the red query cells in the same space as the reference cells behind them. And then we can do cool things with this, like for example, if there are any annotations in the reference, like cell types, we can look for each query cell at the nearest reference neighbors and be able to transfer the annotations from the reference cells to the query cells. And so for that PBMC example with the three technologies, you can see here that we compared Symphony to a gold standard of running de novo integration on all the cells together. But you can see that Symphony does a good job of being able to integrate across the technology variable while maintaining the cell type differences. And so we really designed Symphony as a general purpose tool. And so I want to kind of show you an example where we have several examples in the paper kind of arguing that the resulting coordinate space that you get from reference mapping gives you useful information than the query dataset alone. So here's one example where the query, where the reference literally has a type of modality that's not present in the query dataset. So here we have an example where we're mapping RNA-only cells onto a multimodal reference that was measuring both RNA and protein. And by mapping onto multimodal references, we can leverage rich information that might not be present in the query dataset. And so as a background for this example, recent technological advances have let us be able to do things like simultaneously measure RNA and surface protein from the same cells using oligonucleotide tagged antibodies. So this is site-seq. And so for this analysis, the way we set it up is we have an atlas that was generated using site-seq, so containing both RNA and protein. And then we can try to map in a query data set that only has RNA information. And so here, the pink cells are the query cells. We map them into the reference cells in gray. And then what we can do is then, for each query cell, look at its nearest reference neighbors and try to impute the unmeasured protein expression for the surface proteins. So for this example, we use this data set of circulating memory T cells from a Peruvian tuberculosis cohort that was assayed using CiteSeq, so RNA as well as 30 surface proteins. The full data set includes 259 donors across about 500,000 cells, and we did an 80-20 donor split to create the reference and query data sets. And then we define this coordinate space using a method called CCA, which you can think of as similar to PCA, except for it can combine both the RNA and protein information. And so here's an example of surface protein measurements for CD4 and CD8 in these T cells. And so what we did is we built a reference using both the mRNA and protein information in CCA. And then we mapped in the query cells using their RNA data alone. And then once the query and the reference are in the same embedding space, we can predict the protein expression for each query cell based on its nearest reference cells. So we did this across the 30 surface proteins and found that there's very good correlation between the predicted surface protein measurements and the actual true measurements. And here's 3 examples of proteins where we have the ground truth measurements and then the symphony predicted measurements from the transferring from the embedding. Great. So now that we've talked about how to measure HLA expression in single cells, and we've talked about how to integrate multiple data sets with Symphony, we can put it all together to now understand our question of HLA EQTLs. So if we go back to our main study where we have the 4 different data sets, we next sought to map EQTLs for the classical HLA genes in myeloid, B, and T cells. So what we did was an analysis per major cell type, where we created pseudobulk profiles, where we combine all the cells from a given cell type and individual together into a single measurement, and then did this analysis across samples where we tried to find associations between the expression of the HLA genes and genotype for the different 12,000 SNPs. And we combined all the cohorts together into a single linear model and regressed out covariates. And so by doing this, we get a plot that looks like this. So this is a Manhattan plot, where here each dot is a test between a genetic variant on the x-axis in the MHC region and testing for its association with HLA gene expression for the different genes, which are the different colors. And so you can, the y-axis is the strength of that association. So here I've labeled the variant that has the most significant association with HLA expression levels for each gene. And so if we just take a look at one example, we'll use this HLA DQA1 example for myeloid cells. The top variant was RS310, the SNP here. And so there are 2 versions of the SNP. You can either have a C or a G. And so what we found is that with increasing dosages of the G allele, individuals tend to have much higher HLA DQA1 expression. And this effect was consistent across all 4 data sets. Interestingly, this variant also commonly co-occurs with this HLA DQA1 0301 allele, which has been previously associated with type 1 diabetes and celiac disease. And so what we think is going on is there could be both coding effects or, in this case, also regulatory effects that increase the expression level for DQA1 that could be potentially playing into how the allele is leading to disease. And so to take a look at where this variant is located, you can see it's an intergenic SNP in between HLA DRB1 and DQA1. And we can do things like condition on that main signal and see whether there are conditionally independent signals that are independent beyond the main effect. And so when we do that, we condition on this SNP, there's another independent association with this RS927 SNP that's closer to the promoter of the gene that's also associated with HLA DQA1 expression. And so by doing this, we can find EQTLs that are dependent at the level, context dependent at the level of major cell types. So here's one example where this EQTL RS927 is the lead SNP for HLA DRB1 in B cells. And so you can see a very strong effect for this EQTL in B cells. However, the effect becomes weaker when you look at myeloid cells, and the effect is absent when you look at T cells. So this is kind of showing why it's really important to look at cell types when doing EQTL analyses. And then we can also look to find dynamic EQTLs that are dependent on the cell states within a cell type. So basically everything up until this point, we could have technically done with bulk RNA-seq had we sorted out all these cell populations independently. But now we're trying to be able to capture dynamic regulatory effects that change across fine-grained cell states within this example for T cells. And so biologically, what might be happening when we see these dynamic effects is that it may reflect cell state-specific transcriptional machinery. For example, transcription factors that bind to specific regulatory elements only within certain cell types or cell states. And so the previous analysis I showed you was with a pseudobulk analysis where we collapsed all the cells per sample and cell type into a single measurement. But now I'll show you how we find these dynamic effects using single cell resolution models. Where we, in this case, we model every single cell individually in our model. And so a very important question when doing single cell EQTL analyses is to be able to, how do we define our cell state variable? Because you could do this one of many different ways. For example, you can imagine clustering your cells and then using that discrete label as your cell type definition, cell state definition. Or you can imagine using more continuous definitions of cell state. For example, using principal components or CCA or pseudo time to be able to order each cell along a continuous axis and use that to test for cell state dependent effects. And so in our study, we wanted to be able to define these continuous cell states in a common way across all these different data sets. And so this is where we found that this to be a very good use case for reference mapping. So the idea here is we took the two tissue data sets and then integrated those cells there using PCA and Harmony. And then we mapped in the PBMC data sets into the same harmonized principal components or HPCs using Symphony. So here's an example of what that coordinate space looks like with myeloid cells. Here we have the 4 different data sets, synovium, intestine, and the 2 PBMC data sets. Now all the cells from all the data sets are in the same space. And so we can look at visualize HLA class 1 and class 2 expression across the cell state landscape. And then if we look at these cell annotations that come from the different studies, we can make sure that our reference mapping is aligning the correct cell states together. So for example, dendritic DC2 dendritic cell, classical dendritic cell 2 states are the light blue cells that are properly aligned between both tissue and blood. And then we have the PDCs in the dark blue. The red cells here are infiltrating monocytes and they align well with the blood monocytes from the blood data set. And then the orange cells are tissue macrophages, so we don't expect there to be blood cells that align there. So this sort of fits with our expectation. And then we can do the same thing for B cells and T cells as well. So here we have the coordinate spaces generated for B and T cells. So for example, the major differentiating split in B cells is plasma cells versus non-plasma B cells. And so we see in both synovium and blood the alignment of the correct cell states. And then in T cells, we have all these different T cell states that are aligned across the different data sets. And so now that we've defined our cell state variable, we can now find dynamic EQTLs using single cell EQTL models. So here we used a negative binomial mixed effects model where we model the count of each gene in each cell and data set as a function of donor effects, so things like the SNP that we're testing for association with, age, sex, ancestry, et cetera. Also, we add cell level effects like the library size or the mitochondrial percentage. And then here we also add importantly, the cell state variable, which are the top 10 harmonized principal components now defined in the same way across all the different data sets. And we also add interaction terms between the SNP and the cell state to be able to find dynamic effects that change with cell state. We control for repeated sampling of cells from the same donors using random effects. And so by running this model, we can get a plot that looks like this, where here every dot is a test for the lead EQTL in that cell type and data set, and testing basically for how dynamic the EQTL effect is. So what came out of this is we noticed that, so the y-axis here is showing the significance of how dynamic or cell state dependent this EQTL is. And so what came out of this is that we saw that for across all the different cell types, interestingly, HLA DQ genes, so DQA1 and DQB1, tended to be very cell state dependent. So I'll show you now 2 examples of that. The first example is for HLA DQA1 in T cells. So here we have the T cells from the synovium data set. And what we can do is from our model, we can estimate a total EQTL effect size for each cell by taking the main genotype effect as well as its interaction effects with all the different cell state variables. And that way we can color each cell by basically how strong is this EQTL effect happening in this cell. So if we look across these blue cells, we see that the EQTL is very weak in these cells, has a weak effect, whereas in the orange cells, the EQTL has a much stronger effect. And we can do this in the other data set as well, the PBMC blood data set, and show that the effect replicates, where in the blue cells here, the EQTL is very weak, whereas in the orange cells, it's a much stronger effect. And then we can map this onto the different types of T cells, the T cell states, which are the different colors down here. And so we can kind of see which cell states have the stronger effect and versus weaker effect. And so here on the right, I'm plotting each cell state. The x-axis is the mean HLA-DQA1 expression in that cell state. And then the y-axis is how strong the EQTL effect is mean across all the cells for that cell state. So you can see in both synovium and blood, the cell states with the highest EQTL effect are gamma delta T cells, CD4 cytotoxic, and CD8 cytotoxic T cells. And it's interesting at all that these T cells express HLA class 2, and so it's even more interesting that they're somehow dynamically regulating their expression across cell states. And so now here's another example, this time for HLA-DQA1 in myeloid cells. So here's our myeloid cell landscape across synovium, where we have the blue cells have the weaker EQTL effect. So these are actually dendritic cells, and you can see that they actually have high expression of HLA-DQA1, but the EQTL effect is weaker. And then in the blue cells, the orange cells are the monocytes. Here we can see a stronger EQTL effect for this EQTL. And we can show that this replicates in blood as well, where the blue cells have a weaker effect, whereas the orange cells have a much stronger effect. And here we have the opposite trend as with T cells, where cells that have higher expression of DQA1 tend to have lower genetic regulation, whereas in the T cells, it was cells that have higher expression tend to also have higher EQTLs. Great, so to summarize what we have talked about today, we set out to understand the HLA genes, which are very important for immune-mediated diseases. And then we had to develop two new methods to be able to do this properly in single cells. The first is single-cell HLA-PERS, which is a personalized pipeline to quantify HLA expression. And then the second is Symphony, which is a general-purpose tool for integrating multiple data sets using reference mapping. By combining four cohorts, we created a cross-tissue atlas of HLA expression and mapped EQTLs for HLA genes in diverse immune cell types. And then by using single-cell EQTL models, we found dynamic HLA regulation and pinpointed their cellular contexts. And we think that this regulation of HLA expression levels could contribute to biologically important and potentially disease-relevant variation amongst individuals in terms of their immune response. And in terms of the two methods we developed, they're both available on GitHub. And although I described them to you today in terms of this HLA EQTL study, I wanted to point out that they're broadly applicable to many other questions and goals. For example, we've used reference mapping to be able to try to do things like compare across species, defining different trajectories and adding additional modalities like potentially ATAC-seq. And then for HLA expression, using our personalized pipeline is very important, basically, in any case when you need to accurately measure HLA expression levels, which could be very relevant for cancer and transplantation contexts. And then we also think that our meta-analysis strategy for our single-cell EQTL analysis, where we can combine data sets together to improve power and compare across the data sets, is going to be useful not only for HLA genes, but also for other genes that people are interested in. Great, so I think that's the end of the scientific content. And I have a lot of people to thank, so I'll move on to the acknowledgements. So first, I wanted to thank my advisor, Soumya Raychaudhuri, who's just been such an amazing influence and mentor and supervisor, everything during my time in the lab. He's always so on top of it in terms of both technical and scientific feedback, but also like life advice and things when I've run into different types of challenges throughout my time here. So thank you, Soumya. And then I also wanted to thank, beyond Soumya, the larger lab community, which has just been such a wonderful environment to do research in. So this is our lab retreat photo, and then here's some of us from ASHG last fall. I especially want to thank the grad students in the lab, Katie, Lori, Caitlin, Aparna, Nia, and Anika. And here's a picture from when we were carving grad students as grad students. And in terms of individuals who've contributed a lot to the projects I've mentioned, I wanted to especially thank Ilya Korsunsky, who was my initial mentor during my rotation in the lab and helped guide me throughout the Symphony method and analysis. And so that was extremely helpful. And also Aparna Nathan, who collaborated with me on the memory T cell example I showed you. And then in terms of the HLA work, I wanted to thank Yang Luo and Sari Sakawe for introducing me, helping me get my bearings in the field of StatGen, and then also Amber Shen and Sid Gurujala for their many contributions and enthusiasm and hard work on this as well. And then for For this third project, I didn't have time to tell you about today, but we tried to do some analysis where we compared cell states across species. And for that, I worked with Paul Hoover and David Lieb from Neha Cohen's group, and they were just wonderful collaborators to work with. And here's the full list of various lab mates and collaborators who contributed to these 3 different parts of my thesis work. And then I also wanted to acknowledge members of my PQE, DAC, and defense committees, especially Paul, who served on all 3 of them, members of the, the big program administration, and members of the HST, Health Sciences and Technology program, as well as the Harvard MIT MD PhD program, faculty and staff who've been helpful in terms of navigating the many different aspects of graduate school, as well as the NIH for funding. And then finally, I wanted to thank a lot of friends and classmates, both near and far, for going on many adventures together in and around Boston, including skiing and eating a lot of food. And I also wanted to thank Jason, my partner, who's just been a really great support system for me during my time here. And then finally, I wanted to thank my parents, my mom and dad, for being amazing role models and for all of their love and support throughout my life. And great, so we have 5 minutes for questions. Thanks so much. Great. Any questions? I'm told we can use these mics, so.